So in the next block we will see these split unit designs. So split units are designs in which one of the factors is hard to change, is said to be hard to change. So let's say that we have an experiment with two factors, and one of them requires large experimental units, while the other one requires small ones. Additionally, the second factor can be applied to small portions of the experimental units of the first factor. So for instance, we're investigating the effect of light and diet on the growth of mice, and the experimental unit for the light factor is the whole room, because it is where you can change your light. All cages will receive the same treatment, that is the number of light hours, for instance, and the experimental unit for the diet is the cage. All mice in the same cage receive the same treatment. So this is these experiments in which you have two factors, the second one kind of nested into the first one, are called split unit designs. And let us call P the factor with large units, so this would be the light, and Q the factor applied to small units, this would be the diet. And let us assume that a large unit receives treatment PJ, and it is allocated in the ith block. So from the point of view of the large unit, so we have our measurements that would be the average of all the animals within the same room, would be an overall mean plus something that depends on the block. The block would be the week or the period in which you have done your experiment. So or the center where you have done your experiment. So it is a mean plus the block plus the treatment plus some noise. And let's say that the small unit receives the treatment QK. So for a particular cage, the observation will be an overall mean plus something that depends on I and J. So what is I? I is the block and J is the treatment in the large factor. So this is block and center, let's say, plus something that depends on the diet, plus something that depends on the interaction between the light and diet, not the center. So the blocks, remind that we are not interested typically on the effect on the interaction of blocks, because otherwise they are not blocks, and then plus some noise. And you can easily imagine that this translates into some least squares system matrix, and also this translates into a system matrix for this least squares analysis. Both models can be integrated in a single model. So a measurement for a particular block and light treatment and diet treatment will be an overall mean plus something that depends on the block plus something that depends on the light plus some noise that depends at the large level, so at the level of room, plus some the contribution, the main effect of diet, the interaction between light and diet, and then some noise that depends on the specific cage. And yeah, so here we have the relationships between the different variables that we had in the first decomposition and the variables that we have in the second one. So yeah, so let's say that you are investigating the effect of 5 irrigation systems, that is the large unit factor, and 3 rice variants. So you can do it in this way, for instance. So you can design, you can have multiple blocks. The blocks are different plots where you will apply where you will do your experiment. And then in the first block you have this irrigation P2 with Q1, P2 with Q3, P2 with Q2. So by columns, you apply the same irrigation system, and within the columns, you split it, and you have the treatments for the second factor, which is much easier to change. That is the rise variance. And this idea that when you repeat your experiment, you shouldn't simply repeat this one with the same structure in a different block. So you should randomize the columns, and also you must randomize within the columns. So for instance, here in P2 we have Q1, 3 and 2. Well, now we have 2, 3 and 1. So within the blocks you should also randomize. You can nest several variables, each one requiring an increasingly small experimental units. So you might have multiple blocks, and then you have 3 factors, P, Q and R, and each one is larger than the next one. So for instance, here you have P3, P4, P2, P1. Here you have P4, P3, this is a mistake, no? This is P1. So P4, P1, P3 and P2, and within the blocks you have, within each one of the treatments, you have a larger, so just to put an example, P3 could be the number of light hours, and then on each one of the shelves of the animal house you might apply a different treatment, Q. So I don't know, different ways to ventilate the rack, or I don't know what, and within each one of the racks you would apply a different diet, let's say. And so this would be, this is they are called split unit designs, because you are nesting multiple variables. We have also these crisscross designs. If both treatments require relatively large experimental units, we may apply one of them on the columns and another one on the rows. So here we have, in the columns we have all the time P2, P5, P3, and in the rows we have Q1 all the time, Q3 all the time, and then when you repeat it in multiple blocks, then you somehow randomize the combination of factors. So this would be all for split unit designs, and see you in the next video.